The good news is that yesterday, those of you that were here, I depressed you all, and I'm sorry. I told you about taking beautiful Appalachian mountainsides and turning them into surface mines. So to pay you back for that, I'm going to tell you today a story in the opposite direction of taking agricultural landscapes and converting them into wetlands. This is, actually, let me go back one second. This is a large 1,000-acre agricultural field that I visited back in 2005, shortly after starting my position at Duke. And you might be able to see corn stalks and corn cobs, really flat coastal plain landscape, old agricultural field. And I was invited there by a group of private investors that were going to turn this into a wetlands mitigation bank. And I've been working on this site ever since. And this is what it looked like 1 year after I visited it. And this is what it looked like in 2010. Yay, it looks better, right? It's really pretty. It's a really pretty place. It went from being a corn-soybean rotation field to being a restored wetland, I think, well on its way to eventually becoming a cypress white cedar stream swamp. Pretty interesting place. And of course, the same kinds of questions that I talked about yesterday, the same sort of overarching questions are, what are the consequences of this major change in hydrology and vegetation and the way this land is being used on the export of materials from this landscape? So I'm going to start off giving you a little bit of context, and then I'm going to walk you through the water quality and the carbon consequences of this change. And at the end, I'm going to talk a little bit about how the changing climate context is in fact making us rethink conclusions we might have drawn from the early parts of the study. So converting wetlands to farm fields. So let's start off with a little bit of context. Between 1970 and 1985, about 750,000 acres of North Carolina's wetlands were converted to farmlands. This is pretty late. A lot of this happened just before the most restrictive requirements of the Clean Water Act went into effect. A lot of really beautiful bottomland hardwood and precocian wetlands throughout the North Carolina coastal plain were turned into farm fields, largely by large corporations. So taking beautiful wetlands in which not much had really been done. There'd been some high grading of high quality cypress wood, but they're pretty hard to work in. So it wasn't until a bunch of Dutch engineers came to North Carolina in 1970 with their expertise in draining and dredging and ditching things that we were able to convert this into farm fields. And so we have this very large extent of conversion. And the area that I'm going to be talking about today is part of what became First Colony Farms, which was at the time the largest farm east of the Mississippi River. And just to give you a sense of how attitudes have changed, because I think it's always interesting to think about this, here's an excerpt from an article that was published in 1975 in Science when this farm was going into the ground. And they say its black soils, which include organic mucks up to 12 feet deep, can produce crops of corn and soybeans as abundant as any in the Midwest. In fact, this agricultural frontier represents one of the most promising major reserves of potentially rich but unreclaimed land in the eastern United States because of the peninsula's high potential. Anyway, it goes on and on. So there's an essay in Science about how wonderful it is that we are reclaiming wetlands for agricultural landscapes. So now we're talking about reclaiming agricultural landscapes and turning them back into wetlands. Really, and that's what would end up in Science today. So restoring farm fields to wetlands, in order to make farm fields out of these wetlands, which are in a very flat coastal plain landscape, you have to, of course, remove the trees, you have to ditch them, and you actually have to pump them to keep the water table low enough that you could have crops grow in them. So it requires a fair amount of constant energy input in order to maintain the farms. So if you want to restore the farm field to a wetland, you can, of course, go in and actually plant trees instead of corn or soybeans, and you can stop pumping. And that's really all you have to do to reflood the landscape. If you just turn off the pumps, you're going to get more water, and you're going to reflood that landscape. And the question really is, okay, so we do that, and we wait long enough, will we get back all these wetland ecosystem services that we really value? Things like flood mitigation, the provision of wildlife habitat, the incredible potential for carbon sequestration in wetland sediments, and these really important water quality benefits, which motivate a lot of the mitigation. So we've been asking in this site, which is the largest private mitigation bank east of the Mississippi, what are the biogeochemical consequences of restoring wetland hydrology to coastal plain agroecosystems? This is happening intentionally in a lot of this landscape and unintentionally in a lot of the landscape as well, because as fuel prices go up or as fuel prices fluctuate, these are pretty marginal soils. A lot of this land is getting abandoned and reflooded, not through active restoration, but through just passive stopping of agriculture. So we're interested in whether the changes in nutrient exports, what are the changes in greenhouse gas emissions, how do those trade off against biomass carbon sequestration, and then as I said, the last third of my talk, I want to talk about the changing climate and context and what that might mean in this landscape. So the first time I went out to this site, I went out with my first two graduate students, and we spent a weekend out there. And my student, Jen Morris, was really the reason why we started working there, because she said, I think it's really interesting. If you take an ag field and you convert it into a wetland, and there's all that fertilizer legacy in those sediments, isn't there going to be a lot of greenhouse gas produced from this landscape? Shouldn't we be worried about the nitrous oxide production? And I said, that's a really good question. So we wrote a couple proposals and got this work funded. But really, it was her impetus that got us started down this road. So we really began thinking about this landscape in the context of ecosystem services, or the services of water quality and carbon storage. I think I need this big stick. And I sort of like, and I'll come back to this a little bit at the end, this diagram from Foley et al. In 2005 in Science, thinking about how natural ecosystems provide a wide range of ecosystem services, and often we're converting those natural landscapes into some sort of working landscape where we're optimizing on one trajectory. So in this case, we're looking at making crops in an ag field. And then we might be able to restore these landscapes so that they could provide more services. It's sort of an interesting juxtaposition here. We have a river here, and here we have a working ag landscape, which is a little bit confusing to think about. But let's just think about this water quality regulation, this carbon sequestration components of this story today, which is what we were interested in. And whether there are true trade-offs, or whether it's all, are we talking about all benefits, or are there some trade-offs in this restoration process? So at this point, before I go any farther forward, let me acknowledge my collaborators on this project and our funding sources, and particularly acknowledge Marcella Ardon, who was first a postdoc with me and is now a new faculty at East Carolina University, and Jen Morse, who was my student and is now a postdoc at the Carey Institute with Peter Groffman, who really got us started down this road, and you'll see their name at the bottom of a lot of the graphs that come from their work. So let me introduce you to the specifics of our project. This is a privately owned, as I said, 1,000-hectare mitigation bank. And we focus most of our energy on this 440-hectare agricultural field. So to give you a little sense of scale, it's a mile between each of these rows. It's a really big site. It's kind of hard to wrap your head around it. It's really convenient from the standpoint of a biogeochemist, because we like to put things in boxes. There are two constrained inflows where water comes into the site through pipes, through culverts. And there's a single constrained outflow where water goes out of the site through a culvert. It's surrounded by preservation wetlands, so from the standpoint of dispersal of organisms, fish and trees, et cetera, it's a good place to do restoration because you've got a lot of incoming colonists. It's very flat. There's a less than 5 meter range in surface elevation, and you're going from like 2 meters below sea level to 3 meters above on the high hills of this landscape. And it's confusing from a hydrologic perspective because it's a freshwater system and has wind-driven tides. So when wind blows up against the coastal plain, blows across the sound towards the land, we can back water up through the rivers and then drive water up onto the landscape. So sometimes water flows backwards, and that makes things kind of confusing. And it's not a lunar tide, it's sort of a random wind-driven tide system. So the timeline for this site was that it was a wetland of swamps and a mix of swamps and precocians. It was drained in 1980. First crops were planted in 1985. It went through corn soybean rotation for 16 years before it was purchased by this mitigation, this group of private investors. And this is an interesting case where these are a group of construction company owners who were having to purchase restoration credits from other bankers, and they decided they wanted to do it themselves. And they hired a really interesting and creative consultant who had actually been working for the Army Corps when this happened and had tried to stop the drainage of this wetland. So he had been on this site 20 years before, and he had a real sense of what he thought it should look like in the future. They continued to farm it until 2004. In 2005, they took, the way these are built is there's every 100 meters they dig a ditch to drain water off the farm field that then goes into the canal system. So you have this sort of hummocked field crowns. So the first thing they did was move all those field crowns, dump them into the ditches, plug the canals with big earthen plugs, and they planted 750,000 trees of 12 different species. And the final piece of the restoration was in 2007 when they finished plugging the canals and they turned off the pumps. I'm going to skip over this because I did remember to tell you this. So, okay, well, what happens? So if we take this landscape and we just add water, it obviously looks a lot different, but what are the biogeochemical consequences here? So just to prove that we did in fact increase the water level, this is the water level just at the outflow before the pumps turn off. You see this really spiky pattern here. This is because every day, once the water hit a certain level, the pumps would come on, pump 70,000 gallons a minute, and turn off once you drop below that water level. So you had a very interesting flow regime. Once the pumps are turned off, we got an immediate increase in the water level by 3.5 feet at the outflow, and a huge increase in water across the site. And through a lot of effort, primarily by Marcelo Ardon, we were able to build a water budget for the site based on rainfall coming in and the very complex mathematical requirements of calculating outflow in a bidirectional flow system. But that underpins all of the mass balance we'll talk about. So I want to, because not everyone in this room is a hardcore biogeochemist, I thought I would start off with a conceptual diagram that is unfortunately made in PowerPoint. And I know that Matt has told you all not to do that. Oh, well. So I want to walk you through what you might expect to happen in a coastal plain ag field. Let's think about how biogeochemical cycling works for carbon, nitrogen, and phosphorus in this system. So when you're growing crops on this landscape, you're losing a lot of soil organic matter. In fact, these were old peatlands that were drained, and so there's estimates that in some areas you're losing over 1 meter of soil elevation because you've burned off all this carbon in those stored peats. So you're seeing a net loss of CO2 from soil organic matter, and you don't get any biomass carbon accrual because you're constantly removing those crops off the landscape. So from a carbon standpoint, this is not a retentive landscape. In fact, you're seeing massive fluxes of carbon as that old soil organic matter is being burned off. You're having to add fertilizers each year, both nitrogen and phosphorus, and also potassium, of course. And so you're stimulating nitrogen concentrations in the soils, and we see substantial amounts of nitrate in runoff. As nitrification, these oxic soils that are maintained oxic because you're keeping the water table low, is allowing nitrification to convert ammonium, which is sticky, to nitrate, which is not, which is then leached out of the system, and we see high nitrate losses, which then contributes to coastal eutrophication issues and problems in the canals themselves. Phosphorus, when it's added to all these oxic soils, is really sticky. So it stays on the landscape. You see very little export of phosphorus in these systems as long as the soils are dry. And so you can imagine that each year you add more phosphorus, and the pool of phosphorus in the soil is increasing through time. In terms of the trace gas consequences in this landscape, you can imagine that because as nitrate is leaving the site, you have the potential for both nitrogen gas and nitrous oxide production as that material is being denitrified. And I should actually update this figure because there's a significant amount of nitrous oxide loss in this step as well. So a lot of nitrification-derived nitrous oxide in this landscape. Okay, so what is restoration going to do? Well, you get an immediate benefit that you can quantify, which is known as stop loss. Right, just as soon as you stop farming, you're going to stop that continued soil organic matter oxidation as you reflood those soils. You're no longer adding fertilizers, so you're not adding new phosphorus to the soil pool. And you're no longer adding new nutrients that can run off. So that's an immediate benefit that you get from restoration, regardless of what happens next. When you plant trees, you start to see this positive effect of accumulating carbon in biomass and sequestering nutrients in biomass as well. And you start to see soil organic matter inputs increasing because now you're not taking the biomass off. So all of that root-derived litter and leaf litter can actually begin to accumulate. You can start to rebuild those soil organic matter pools. When you flood the site, there's some interesting implications. Now, this is going to be an ideal situation for promoting denitrification, and that's one of the reasons we build wetlands, right? We want to make anoxic soils where the microbes that take nitrate and convert it to the harmless inert N2 gas that makes up 70% of our atmosphere are going to be happy. And so you'd expect that as you convert a dry agricultural soil into a flooded wetland soil, that we'll see significant amounts of nitrate being converted into N2. Problem is that same flooding of the soil is going to really block the amount of ammonia that can be converted to nitrate because those are obligate aerobes that cannot do their job when soils are flooded. And so you can start to see ammonium accumulating in the landscape and the potential for increased losses of ammonium, which don't have this large gaseous loss component. And a bigger problem is that, as I told you, phosphorus is really sticky when the soils are oxic. It's sticking onto iron minerals in the soil, and once you flood soils and iron is reduced, that phosphate that it's bound to is released. And so you have the potential for this agricultural legacy phosphorus then to be released from the system. So that's a potential negative consequence of flooding. And then, of course, from a greenhouse gas perspective, if we increase denitrification, we may also increase nitrous oxide production as a byproduct of that process. And when we make flooded soils, we may dramatically reduce methane oxidation, so we'll see more methane being exported from this landscape. So this is sort of our starting understanding. And so if you think back to this kind of model, you imagine that, I'm just putting these parallel to the Foley diagram, we're expecting really good things of denitrification for our water quality regulation benefit. We're expecting reduced soil carbon losses as our carbon sequestration benefit. But for each of these lines, you might expect other trade-offs that work in opposition to that benefit. So we might see increased denitrification coupled to increased phosphorus release, which is a negative consequence, right? And we might see reduced soil carbon losses, but what if that's coupled with an increase in greenhouse gas production? So, it's not as simple as water quality benefits as a single cumulative factor. There's going to be underlying pieces of that require you to think about individual solutes separately. So that's where we were sort of starting our research from. But what do we actually do? So I did what every good ecosystem ecologist does when they encounter a system, and I stuck it inside of a black box, because that's what I do. And we, sorry about that. And we used basically weekly samples and stormborne samples of inflow, nutrient concentrations, and water flow and total flow amount from the 2 inflows to the site versus the single outflow from the site in order to build a mass balance for the nutrients coming, the form and the quantity of nutrients coming in and going out, to think about this water quality consequence. So, good news first, very significant attenuation of nitrate pulses. So upstream of our site is an enormous farm that is, for some reason that I don't understand, owned by the Mormon Church. Oh, you should all turn off your cell phones. Sorry about that. It probably is a robocall. I'm really sorry about that. So this is looking in the inflow, which is being basically pumped across off this agricultural field. So we can see that indeed, this agricultural field is losing a significant amount of nitrate during the fertilization period. So we know that nitrate inputs are coming off the agricultural landscape, but here's export from this wetland. So we see absolutely no change in nitrate over the season, despite very large fluxes of nitrate coming in. So the really good news from this, this is from the first year of the study, was a 90% reduction in nitrate export as a result of wetland restoration. Great news. And if we only measure nitrate, which is what is being done in some wetland restoration projects, we could call this a fabulous success. But we were thinking about other forms of nitrogen. And so if we look at ammonium concentrations, you'll note that the situation is reversed. The open circles are the observed, sorry, I should have explained. The open circles here are the observed export. The black circles are our modeled estimate of the pre-restoration flux. This is based on what's coming off that agricultural field and a historic flow data from before the restoration started. If you want to know more, you can read this paper to calculate these flux changes. But what we see is an increase in ammonium export from the site relative to the pre-restoration state. Another way to say this is if you look at outflow ammonium concentrations, they're higher than inflow ammonium concentrations. And we found that actually we saw an increase, we doubled the export of total nitrogen from the landscape, but we changed the form completely. So instead of losing nitrate from the system, we're losing significant amounts of ammonium here in the spring and again in the late fall. Keep that in mind, I'll come back to that at the very end. And we're losing significant amounts of organic nitrogen in the spring. So that's a bit more of a mixed bag than you might think otherwise, because this ammonium, once it's exported, when it gets into an oxic water, can be nitrified and contribute to nitrate loading to the coastal system downstream. Well, what about phosphate? And I've grayed that out because I'm not concluding yet. I wanted to walk you through what's going to happen. Again, remember, in a dry soil, phosphate's going to bind onto aluminum and iron oxides. But once you flood that soil, that phosphates that are bound to that oxidized iron is going to be reduced, and the phosphate bound to that iron will be released and can be exported from the system. And indeed, if we look at reduced iron across our site, so this is inside the black box looking at our sort of sampling regime inside the black box is looking at permanently flooded, intermittently flooded, and never flooded sections, transects across the site. Here's poor water, reduced iron concentrations, and you can see indeed in our wettest sites we have high concentrations of reduced iron, much less reduced iron in the dry sites, suggesting that there is certainly the potential for the release of phosphate bound to those materials. I don't know why that did that, sorry. And so Marcel has a whole lot of data on the phosphorus release from the system, but we do see I should go back here first, sorry. We do see that, as we expected, total phosphorus export increased a lot as a result of this restoration. So a big spring peak in phosphate reduction. We actually increased TP export by 6 times, and most of that was as phosphate, as soluble reactive phosphorus. So we're seeing a dramatic increase in inorganic phosphorus released from this system, purely as a result of flooding it and allowing all that bound phosphorus to be released from iron. And so that's a big deal. Now, this is undoubtedly a transient effect because we're basically working through this fertilizer legacy stock in the system. We can't yet detect a significant decline in soil phosphorus. Here again, looking at the driest parts of the site, these intermittently flooded parts of the site and the wet parts of the site, total soil phosphorus content over a 2-year period, versus pre-restoration in black, 3 years after restoration, and another year following that. You can see a general decline that is stronger in the wettest parts of the site. So our modeling effort on this suggests that it'll take about a decade to basically work through that legacy phosphorus before we won't see elevated phosphorus release from this site. And of course, there's uncertainty around that, but I won't focus on that right now. So in terms of water quality benefits, it's a bit of a mixed bag. As you'd expect, we do see a elimination of fertilizer nitrate pulses from the system. Clearly, nitrate that's being delivered from the upstream farm is not making it out. That could be because of denitrification, it could be because of plant uptake. But that's coupled with this increased phosphorus, ammonium, and DON export, which should substantially cool our excitement about the water quality benefits of this restoration. Again, the phosphorus issue is certainly a transient one. The ammonium one maybe as well. This may be a question of time, but certainly it's not an immediate benefit. And so I think one of the interesting questions in thinking about how to take this information back to improving restoration is that there are ways to manage the landscape to minimize the time of this legacy effect. So if we don't have to wet it up right away, we can let plant biomass growth sequester a lot of this material before we flood it. And we can talk during the question session about some of the regulatory constraints that make that hard. Okay, so let's go back to our black box and think again about carbon sequestration, this trade-off between biomass carbon accrual and greenhouse gas production. So we have to open the black box to do this. And so we've been working pretty hard to use LiDAR remote sensing estimates to get an estimate of total woody biomass in this landscape. I know some of you in the audience do this kind of work. It's really hard to do with sparse vegetation. And so we have enormous uncertainty bounds around our estimate of carbon storage and biomass in this landscape. But we've done the best we can, as you would expect. If you plant 750,000 trees and leave a large nutrient-rich wetland alone, trees grow. And that works great. So we certainly see carbon sequestration in the landscape. But we want to understand how much greenhouse gas emissions might offset that. So we set up, as I showed you before, these transects across the site, heavily instrumented with wells for measuring water quality throughout the soil profile, permanently installed redox probes, soil moisture probes, and gas wells where we could periodically measure the gas fluxes. Just to give you a sense of the difference in the hydrology between those 3 sites, here's continuous water level recording data for one transect where you can see here the dry site, which becomes flooded only after a couple of rains and quickly drains. The intermittently flooded site, which is permanently flooded for about 3 months of this year. And then the permanently wet site where water is always above the surface. And so this is the very difficult work of Jen Morris's dissertation. And it just came out last month in EcoApps if you want to learn more of the details. But so looking across these 3 different hydrologic categories, Jen measured methane, nitrous oxide, and CO2 fluxes over every other month for 2 years, and used that data to estimate the total CO2 equivalents produced from greenhouse gases. Now, remember, our question here is whether or not we're promoting enhanced greenhouse gas emissions from this site relative to its agricultural past or relative to natural wetlands. And I'm really happy to report that for this system, that certainly doesn't appear to be the case. So here are CO2 equivalents. Here are the three categories in our restored wetland: the dry, the intermittently flooded, and the permanently flooded site. CO2 is the big part of this bar, and then we've got nitrous oxide and then methane. Oh, God, I'm so sorry. I'm really sorry. There we go. Wrong button before. Okay. At least it's a cricket, right? It's like ecology. So we're comparing this to similarly collected data for two nearby preserved wetlands that have not ever had any kind of agricultural ditching or draining or planting, and that upstream agricultural field that the Mormon Church was kind enough to let us sample on. And you can see that we don't see a substantial increase. And in fact, the most nitrous oxide we see is from the dry agricultural field. It's mostly coming from nitrification. And we have substantial reduction in the nitrous oxide and methane fluxes in our restored wetland. And those fluxes are on par with what we see in our preservation freshwater wetlands. So at least for this system, really no evidence of enhanced nitrous oxide losses, which is really encouraging. And I think the methane is interesting because this has become a relatively carbon-poor system through all the soil oxidation. So there may not be a ton of carbon there to be supporting large amounts of methane flux. So let's think about the ecosystem benefits versus the ecosystem costs of this project so far. So in terms of benefits, we clearly have, I would say, better habitat. Now, what are my metrics of that? There's not many things living in a cornfield, and this site is now full of fish and a lot of waterfowl. So certainly I don't have a quantification of that benefit, but certainly the habitat quality is better. There's many more species living there than were living there when it was a corn and soybean field. We've seen a reduction in nitrate, an increase in biomass carbon. We've stopped loss, the soil organic matter oxidation, and we've actually reduced greenhouse gas emissions from the site relative to its agricultural past. And we're trading that off against these fertilizer legacy exports that we expect to last somewhere up to a decade. So I think this project is a really interesting project, and I think it's largely headed in the right direction. I'm pretty confident that in a decade, this is going to be a really beautiful place. And already it has tremendous benefits over its agricultural past. It's providing a benefit to downstream ecosystems in removing fertilizer that's coming from that upstream agricultural site. So I think the balance sheet here looks pretty good for this restoration project. And in part, that's because it's really big. It's a very large restoration project. It takes up an enormous chunk of its the total watershed, and it has the capacity to really do a lot of work. But I want to get into what I think are the really interesting things emerging from this, and also give you a caution that you should look at your data as you're collecting it so that you can be better prepared for data, for surprises in your data set. So let's talk about this changing climate and context for this landscape. So our site is right here near Columbia, North Carolina, and I'm showing you here predictions of sea level rise for the coastal plain of North Carolina. Let's see, this is the low climate change estimate, and the high climate change estimate is in dark burgundy. Really, no matter what, sometime in the next century, this site is going to be underwater. It's going to be part of the sound. So that's interesting, and it means that our site is headed towards a seawater fate, right? And we should have thought about that a little bit. But there had been flap gates installed in this site, right? They were preventing any water from ever exchanging with the sound because you were constantly pumping water downstream. You had a dam built by the pump gates. So we collected data on conductivity every week for a year and a half before we looked at it, which we really shouldn't have done. This is here the water level for our site over the since the restoration began up through this past summer. Actually, Marcella got it all the way up to this month. In gray, and here is conductivity. And we really weren't expecting this. So conductivity is going up to brackish water concentrations in the middle of these summer droughts. And we had two really major centennial droughts in the last 10 years in the site. So these were times when actually there was huge peat-burning fires in the coastal plain of North Carolina caused by lightning strikes. And whenever we saw those really big droughts, we saw saltwater intrusion into our site. This is a surface water phenomenon. The groundwater is not brackish at all. It's purely fresh. And it's water, basically chemicals diffusing up gradient from the sound into our site. And so in really dry summers, the sound gets much saltier, and those salts mix upstream. It's facilitated by wind mixing, but pretty big change, right? And we'd done all this biogeochemical consequence work without even thinking about the fact that salinity was changing in the system. So we have to ask ourselves, how will saltwater intrusion alter the answers to the above questions? And this is important not just for our site, but for this whole area of the coastal plain. It's these ag soils that are headed towards being part of the sound. So this gets really complicated because now we already talked about how we've got this nitrate story, increased nitrate coming in from the agricultural systems, but we've got a bidirectional flow system with increased sea salts coming in upstream, moving upstream, bringing chloride, magnesium, calcium, and sulfate, and we're sitting in a site with variable soil iron content. So we are working on coupled carbon, nitrogen, oxygen, iron, and sulfate cycling. We're going to spend the rest of the talk discussing this, and there'll be a quiz at the end. No, I'm kidding. So I'm going to get a little bit intense here because this is hard, right? We actually don't know how to do this very well. We as biogeochemists are spending a lot of time talking about linking carbon and nitrogen cycles and maybe phosphorus. But to understand what's going to happen in this landscape, we have got to bring sulfate on board, and we've got to bring iron on board. So sulfate is a big player in actually both facilitating and depressing denitrification, which makes it really complicated to figure out what it's going to do. And iron, as we've already talked about, is really important in phosphorus dynamics, and also interacts with sulfur in ways that make it kind of hard to predict what's going to happen in this landscape as a result of these complicated biogeochemical changes. So our approach to this, and this is work that Ashley Helton, who's a postdoc in my group, is spearheading and is in close collaboration with Jeff Poole, who's a agent-based hydrologic modeler working at Montana State. We've been building a thermodynamically based model that links all of these cycles together with the common currency of free energy dynamics. And I'm not going to ask you to memorize this either, but basically the idea is you have to be able to simultaneously account for all of these things. And we made the argument in the proposal that finally got funded 3 rounds in that you cannot do this without simulation modeling because it's too complicated to keep the hypotheses straight in your head and make sure that they are all internally consistent. And so we're using an agent-based modeling approach that basically says, and you can't read this and that's okay, here are all the metabolic processes that you could do to degrade carbon in this landscape or to get energy out of reduced sulfur compounds. And so there's a wide variety of metabolic strategies that can be employed, and they each have a different free energy yield. So this is the classic redox ladder that you either know intimately or you've heard of at some point in your past. So our model is really simple. We assume that microbes as a group will do whatever it takes so that they can optimize biomass accumulation in each time step. So you're going to use your energy, use whatever energy you can get out of the substrates provided to you to make new biomass, or to lose the least biomass possible if you're only able to supply your respiratory requirement. The pathways thus depend upon the relative free energy yield, so there's a competition among pathways. But we think about this as just one giant bacteria that can do everything, doing whatever it works best for it at the time. We make sure that the respiratory requirement has to be satisfied prior to biomass growth, and assimilation is constrained by biomass stoichiometry. So we link cycles in 2 ways. One, through relative free energy requirements and the interactions of molecules in these reactions, and also through biomass stoichiometry, because you can't build biomass based on the nitrogen and carbon availability without also accounting for being able to have the right phosphorus availability. So energy may get wasted if you can't build that biomass because you're limited by a nutrient. Okay, so what does this mean? How do we work with it? It's really cool. We have a model now that people can use in Excel, and we'll be sharing it soon so people can start playing games with it. But let's just take your classic redox ladder that you'd read about in the textbook would only really include aerobic carbon oxidation, what most heterotrophs, including us, do, denitrification, you'd have nitrification, you'd have methanogenesis, and you might, if you wanted to, add these processes that we're excited about now, like dissimilatory nitrate reduction to ammonium and anammox. So if you just did carbon, nitrogen, oxygen, you could run a model predicting methane production across an oxygen gradient. You could add in sulfate reduction and iron reduction in your second scenario, and you could also add in a whole bunch of anaerobic methane and sulfide oxidation processes. You don't need to understand all of this in order to be able to see in this diagram the amount of methane that was going to come out of the system is going to change a lot depending on whether you use just a carbon, nitrogen, oxygen assumption or you add iron and sulfur into this dynamic, right? So this means predicting what's going to happen in this landscape means we can't do it without sulfate. And we also can't do it without iron. And so the idea is that we generate variability because as we run optimization runs for this, many, many iterations of optimization runs, there's some variability in what actually happens in each time step. And we also can generate patterning in the landscape, both in the vertical and the lateral dimensions, because we can set up nodes which all use the same exact rules but exchange materials using hydrologic models. So now we can actually recreate the vertical zonation that you see. This is actual real field data from peepers out in the field for reduced iron, sulfide, and sulfate. And so we can begin to use these models to suggest what our best guess based on free energy yield should be happening out in the landscape. Okay, and I'll go ahead and scoop myself a little bit and tell you it doesn't work for one of our major questions, which is exciting because the model is really not the goal. The model is our null hypothesis, our best understanding of the system at this point. So we've been doing, and I'm just going to talk about this one experiment for the end of the talk instead of also our field work on this point. We take intact field soil cores from the Timberlake site, we bring them back to the lab, and we expose them to just water, deionized water, seawater, homemade seawater from which we remove the sulfate and replace it with chloride, so it's got the same ionic strength but no sulfate in it, and sulfate alone at the same strength as in seawater. And then we also manipulate hydrology. So we'll see a couple graphs that look like this. Here on the left, we got a site where we're keeping the water table below the surface. Here is a place where we're keeping the water table above the surface. And so I'm just showing you the treatment effects here. This is chloride. We have red is the DI, blue is the artificial seawater, green is the artificial seawater minus sulfate. You can see chloride goes up in both of our artificial seawater treatments and does not go up in the sulfate treatment. And sulfate goes up in our two sulfate treatments but does not go up in the artificial seawater minus sulfate. So I'm just showing you that to show you that our treatment is effective. So remember, our model suggests, and our thinking about these landscapes suggests, that as we add sulfate to this landscape, we're going to see suppression of methane production, right? So here's methane when we don't have any sulfate included in our model. Here's methane production when we add sulfate to the model. It's not going to happen. We're going to suppress sulfate because sulfate reduction is really high. It's a much higher energy yield than methanogenesis. So what happens? The first thing that's going to draw your eye here, but it's not important, is that there's not a lot of methane coming out of dry soil, so don't worry about that. That's not surprising. What is surprising is let's first look here at red is the DI water. So that's what's happening without any chemical alteration. So we do see methane flux. When we add sulfate, here in the yellow, we completely suppress methanogenesis. Okay? So that's really interesting. We suppress it for a very long term. This is an 18-week experiment. But when we add artificial seawater, we enhance methane production. We enhance it less if that artificial seawater contains sulfate, but we enhance it a lot and we enhance it in a sustained way. So we're seeing an increase in methane production from this landscape that lasts for 10 weeks until the end of the experiment, which is really surprising and interesting. And we don't know why at this point. We have a couple of ideas, and I'd love to hear your thoughts as well. There's a few other researchers who are working in coastal plain landscapes starting to see saltwater intrusion who are seeing the same thing. We have a little bit of tantalizing evidence that it's happening in the field as well, and we can replicate this in slurry additions. So it's interesting. We can replicate it in slurry additions that are done with sodium chloride, so it's not necessarily a cation issue coming in with those sea salts. That's about as far as we can get thus far. But I think it's exciting. We have an exciting model failure at this point. Unexplained biogeochemical phenomena, which is exactly what we want. We want the model to fail in interesting ways. Another really interesting thing, again, making us go back and rethink our data. Remember way back I told you there was this big ammonium release after restoration, and it happened in the late fall. In the late fall is when we have saltwater intrusion during these big droughts. Well, how do you measure the ammonium content of soil in the lab? You add salt, right? So not really shockingly, when we add salt to our soil cores, and both the blue and the green, remember, are our saltwater additions, we see dramatic increases in ammonium concentrations. So we're basically taking those cations in salt water and we're exchanging them on cation exchange sites for ammonium. So just as we would do in the lab if we wanted to extract ammonium, when we put salt water into these field mesocosms at much lower concentrations than you do in the lab. We see incredibly large increases in ammonium leaching. So no doubt a part of that early story about ammonium leaching from our site was because of saltwater intrusion. And so that's exacerbating this legacy release. And so I'll show you that here. Here's ammonium concentrations at our site. This is in the middle of that big saltwater intrusion event in that first year. And so we see this dramatic increase in ammonium loss that at least is partly due to saltwater intrusion. The other thing that's really interesting is the other thing that sea salt cations do, cations with 2 positive charges stick to organic matter, and there's 2 charges, so they can stick to 2 pieces of organic matter at a time. And so one of the reasons that there's not a lot of DOC that makes its way from the land to the ocean is because as soon as DOC from a river hits the ocean, the DOC flocculates with the sea salts and falls to the sediments. And the same thing happens when we have saltwater intrusion in our site. So here's DOC concentrations in black here versus conductivity. And so when we get this saltwater intrusion coming in, we have this dramatic reduction in DOC concentrations going from 60 to 80 milligrams per liter down to sometimes below 10 milligrams per liter. Huge change. And this may be part of the methane story. We don't know yet. That's one of our hypotheses, that by bringing a lot of DOC out of the water column and basically attaching it to the sediments, we may be fueling methanogens, or maybe we're changing the amount of oxidized sediment that's a place where methanotrophs can live. So some pretty interesting and complicated stories that are physical chemistry changes, cation exchange changes that suggest to us that our free energy model is not going to be enough. We're going to have to think about these other aspects of the changes as well. I want to make sure we have plenty of time for questions, so I'm going to stop there with the conclusions that coupled element cycling has got to expand beyond carbon and nitrogen, and particularly in the coastal plain landscape, but also all kinds of other landscapes. I feel that I'm being attacked by sulfate on all sides because it turns out sulfate increases with urbanization. Sulfate in streams increases with urbanization. If you were here yesterday, I talked about how sulfate from coal mining is a huge change in the chemical signature of rivers. So sulfur is really important, and I think we've ignored it for far too long. Free energy is clearly not going to be enough. I think it's a good start on a null hypothesis. It's going to be interesting to think about how we can include things like cation exchange and cation bridging into our thinking as well. And of course, these biogeochemical scenarios really depend on hydrology. Whether or not we get increased intensity of drought, the speed with which sea level rises is fundamentally going to change our view of what's going to happen in this landscape over the next 100 years. It's a really complicated and dynamic place. And with that in mind, the idea of restoration in this changing climate becomes sort of silly, right? There's no way we're going back. This whole place is going to be part of the ocean in the not too distant future. And so a lot of the discussions in North Carolina management communities is how can we prepare, how can we adaptively prepare for the movement of saltwater onto these coastal plain landscapes? How can we prevent saltwater from leading to huge flushes of fertilizer legacies from these agricultural sediments into the future? Is there something we can be doing to them now to tie that material up in biomass, to move it off the landscape before we basically just transport it into these very sensitive sounds, which have really important fisheries? And I think there are a lot of interesting questions there. At this point, I probably talked to all of you for far too long. So I'll stop, and hopefully we'll have time for some interesting questions. Thanks a lot. Okay. Any interesting questions? Or uninteresting questions? Oh, lights. Jill.
There was a paper a couple of years ago, and I'm not going to do it justice because I don't remember it, but it was talking about coastal areas that are flooding outside the eastern shore of Siberia, and the methane flux that was coming from there. And they were presenting this as, isn't this fascinating, and is this the future? So I wonder if it's the same phenomenon. It sounds like it must be.
Maybe. I'll have to ask you for that paper, because there's not a ton out there yet. We've done a lot of work on methane production in salt marshes, and a lot of work on methane dynamics in freshwaters, and not a whole lot in the transition zone. So when we were looking for predictions in the literature, it's a little bit hard to figure out what's going to happen. And you know, what's the trajectory? Is it a linear trajectory? Is it some sort of hump-shaped curve? It's really hard to figure out. But it's been repeated in enough experiments in the last year or so that I'm sure this is certainly an issue. And it is a little bit worrying, yeah. I think I might have overwhelmed people with too many elements. I was worried about that. Any other questions? I'm sorry, but we got to move beyond carbon and nitrogen. So it's going to make giving talks really complicated in the future. Yeah.
I'm curious what your next idea is for that model if the free energy is not really capturing what's happening there. You're talking about improving that model. I'm curious what ideas you might have.
Well, so we can use the model to experiment to test some hypotheses, or to explore some hypotheses would be the better way to say it. So we can actually play with the model and say, well, let's put in a function where as we increase salinity, we change carbon availability in a fundamental way. What would that do to the model? Right, and we can include some interactions between ammonium availability and methanogens. There's a couple people suggested some interesting places there that would be worth some additional research. So we can basically, before we even go do manipulations in the lab, we can sort of say, well, is there any way we could add new rules that would achieve that pattern? There may be multiple ways in which we could achieve that pattern. And then that allows you to target your research on the specific mechanism. So it's kind of powerful because the rule set's really simple, and it's easy to add and subtract from that rule set and see what the consequences are for the way we understand the behavior. So it's actually built to fail on purpose and to allow you to have a modular way of bringing new information in or pulling it back out if it doesn't help. Yep.
Does the model have a spatial element such that when you reduce iron to use it as an electron structure, then that phosphorus actually gets removed from the landscape like you're finding?
It will. So right now, in this version, we got dinged twice for being totally ambitious and trying to go all the way to landscape level hydrology, biogeochemical modeling. So this current proposal is just doing the vertical hydrology. But yes, it's an agent-based model. So you can set it up to have as many patches as you want with depth or laterally. And so and it's sitting on top of a hydrologic model. So the node in each time step will move water and solutes and heat between that node and all the nodes adjacent to it. So you can build theoretical landscapes, which is also really interesting. You get pattern emerging from a bunch of patches that do exactly the same thing, which is pretty neat as well, and allows you to ask some different questions about the patterning of biogeochemistry that may tell us how much we don't know about that, and probably will, right? Yeah.
How does the size of the fluxes of greenhouse gases that we're seeing relate to like overall vegetable?
Oh, yeah.
How much of the coastal flux?
We haven't done that yet. That's a really good point. Just take these estimates and scale them up. I'm a little worried about our upscaling right now, and that's one of the things we're working on is, how do you, this is a classic terrible black box approach, is to get an average rate and just scale it up by acre, and really trying to do a little bit better job by using at least some statistical relationships between hydrology and soil carbon or something like that to be able to scale more appropriately across the landscape. So I'm not sure, we could come up with numbers. I'm not sure how much, if any, faith I would have in them at all. I guess we have seen, actually I should say, we have seen actually pretty high nitrous oxide fluxes compared to other places that have studied nitrous oxide fluxes in our natural wetlands, which is interesting. And that may be because we're sitting in a landscape that's full of hog farms and has a lot of ammonia deposition. We don't know. That's another place that we're interested in exploring. Joe.
There are a whole bunch of aqueous geochemical equilibrium models.
Yeah.
And they work pretty well. But is the difference that you've added in microbes?
I hope so. Right. You mean just that don't include any sort of biological mechanism at all?
We work with one called Freak C, and we've coupled it to Century. Well, Melanie Hartman has.
But to do trace gas production?
No.
Yeah. So that's the issue, right? That the trace gas production. Right. Oh, it does do trace gas. Does it work?
Well, it's all hypothetical.
Right. So we don't know. I think it's interesting. It'd be interesting to take those two different models and play them off against each other. What I like about this model is it's really simple and I can understand it. I can't code it, but I know exactly what it's doing, and I can do the rules myself. And I can take a toy version of this model and work in it in Excel, so I can play with it, which is really appealing. And so the learning curve isn't as high to be able to actually use it myself. But it would be an interesting thing to compare. Yeah. Okay, I've officially worn you out. Oh, no, one more question. Okay.
Well, I was just wondering if you were doing anything much
Oh, ammonia volatilization. No, we are not doing that. And I think, our ammonium concentrations aren't such that you would expect that to be really important. It's really important in the landscape because this is the coastal plain of North Carolina where most of the pork in the United States comes from. And a good reason for eating local pork because we have huge hog farm sewage lagoons and things like that. And there we definitely have ammonia deposition as a major issue in the landscape. But I don't think we see a lot of ammonia volatilization. I don't know about the ag fields. I don't think anybody's been doing that in our landscape, but you do see that in some places following fertilization. Okay. Well, hopefully that's a little bit more optimistic message, maybe a few too many elements, but I wanted to leave you with something a little bit happier. This, I think, is an interesting future story. Thank you for your attention.